Hello and welcome to our channel. Imagine a world where the cruel lottery of genetics no longer holds sway, where devastating hereditary diseases like cystic fibrosis, which relentlessly attacks the lungs and digestive system, or Huntington's, a neurodegenerative disorder that slowly steals a person's physical and mental abilities, could be erased from a family's lineage forever, not just managed with a lifetime of treatments. What if we could become the architects of our own biological destiny, reaching into the very blueprint of life, and correcting the typos, the misprints, the faulty instructions written into our DNA before they can manifest as suffering? This isn't a far-off dream or a page from a science fiction novel. This is the reality we are stepping into, thanks to one of the most significant scientific discoveries of our time. Get ready to discover the universe's ultimate DNA editor, CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Might sound complex, but its origin story is a beautiful example of nature's ingenuity. For billions of years, bacteria have been locked in an endless war with viruses. To survive, they evolved a sophisticated immune system. When a virus attacks, the bacterium captures a small snippet of the invader's DNA and stores it in its own genome, within those CRISPR sequences. Think of it as a molecular mugshot gallery. If the same type of virus attacks again, the bacterium uses these stored DNA snippets to create a guide, known as a guide RNA. This guide RNA then teams up with a powerful protein, most famously one called Cas9. Together they form a search and destroy unit. The guide RNA acts like a highly specific GPS, leading the Cas9 protein directly to the invading virus's DNA. Once the target is found, the Cas9 protein acts like a pair of molecular scissors, precisely cutting the viral DNA and neutralizing the threat. In 2012, scientists Jennifer Doudna and Emmanuel Charpentier had a groundbreaking realization. This bacterial defense system could be repurposed. They figured out how to create their own custom guide RNAs to target not just viral DNA, but any gene in any organism. This transformed CRISPR from a bacterial curiosity into a revolutionary gene editing tool. It allows us to find a specific gene within the vast 3 billion letter-long human genome and use the Cas9 scissors to make a precise cut, cutting out a faulty genetic code. Once the DNA is cut, the cell's own natural repair machinery takes over. We can either let the cell patch the break, which often disables the faulty gene, or we can provide a new healthy DNA template, which the cell then uses to patch the gap, effectively replacing the faulty sequence with a healthy one. The potential was immediately clear, and scientists have already used CRISPR to achieve incredible feats. In the fight against cancer, researchers are supercharging our own immune cells. They edit T cells to make them better at recognizing and destroying cancer cells, a revolutionary approach known as CAR T-cell therapy. They are also using it to treat debilitating blood disorders. For patients with sickle cell disease, a single genetic mutation causes their red blood cells to deform into a rigid sickle shape, leading to excruciating pain and organ damage. In groundbreaking clinical trials, doctors are taking a patient's own blood stem cells, using CRISPR to correct the faulty gene in the lab, and then infusing the edited healthy cells back into the patient. The results have been life-changing. Patients are seeing their blood cells transformed from sickled to healthy, round shapes, freeing them from the symptoms of their disease. And it's not just blood. Children born with a rare form of genetic blindness called Leber congenital amaurosis 10 are now gaining sight. In this therapy, the CRISPR machinery is delivered directly into the patient's eye to correct the mutation in the retinal cells. Imagine seeing the world for the first time thanks to a single precise edit in your DNA. Beyond medicine, CRISPR is also transforming agriculture. Scientists are developing crops that are more resistant to drought, disease, and pests, helping to secure our global food supply. They've created mushrooms that don't brown and tomatoes with improved flavor and shelf life. The power of CRISPR lies in its unprecedented precision. The human genome contains over 3 billion base pairs, the letters of our genetic code. Finding and editing a single gene is like finding one specific house on a map of the entire world and repainting its front door without spilling a drop on the lawn. CRISPR allows us to target these specific genes among billions with an accuracy that was once unimaginable, measured in single letters of our genetic code. This molecular-level accuracy is what makes it such a powerful tool for both research and therapy. We're entering an era where the very concept of incurable genetic disease could become a relic of the past, a future where aging could be slowed and human potential could be unlocked in ways we are only beginning to imagine. But with this great power comes an immense and profound responsibility. The ability to alter the human genome forces us to confront some of the most challenging ethical questions of our time. A critical distinction must be made between somatic editing and germline editing. Somatic editing, which is what current clinical trials are focused on, targets the body's non-reproductive cells. The changes are confined to the individual patient and are not passed on to their children. Germline editing, on the other hand, would involve altering DNA in sperm, eggs, or embryos. These changes would be hereditary, passed down through all subsequent generations, permanently altering the human gene pool. This raises the specter of designer babies, where we might be tempted to move from curing diseases to enhancing traits like intelligence, athletic ability, or physical appearance. This opens a Pandora's box of societal dilemmas. Who decides what is a disease to be cured versus a trait to be enhanced? Furthermore, there's the question of equity. If these revolutionary therapies are astronomically expensive, could CRISPR create a new form of genetic inequality, a world of genetic haves and have-nots? We must also consider the technical risks, primarily off-target effects, where the CRISPR scissors might accidentally cut the DNA in the wrong place, potentially causing unforeseen health problems. These are not easy questions, and they require a global conversation. Scientists, ethicists, policymakers, and the public are working together to establish clear guidelines and regulations to ensure this technology benefits all of humanity, safely and equitably. Despite these challenges, the promise of CRISPR is undeniable. It isn't just changing medicine, it's revolutionizing biology itself. Its applications extend far beyond human health. Scientists are exploring using CRISPR to bring back extinct species like the woolly mammoth, to make coral reefs more resilient to climate change, and to engineer microbes that can clean up pollution. Every day, new breakthroughs bring us closer to a world where the suffering caused by genetic diseases is history. CRISPR truly is the universe's ultimate DNA editor, a tool of immense power and potential. It has given us the power to rewrite life's instruction manual, to correct its most tragic errors, and to secure a healthier, more resilient future for generations to come. We stand at a pivotal moment in human history. The genetic revolution has begun, and its course will be determined by the choices we make today. If this glimpse into the future of genetics amazed you and made you think, please give this video a like and subscribe to our channel. Your support helps us create more deep dives into the incredible scientific breakthroughs that are shaping our world. And don't forget to check out our other videos exploring the cutting edge of science. Thanks for watching, and we'll see you in the next video.